Ladies and gentlemen, welcome to the second class of the selected topics in biopsychology. As you remember, in the first class, we were talking about biopsychology as a division of neuroscience, the methodology of biopsychology, and the principles of the biopsychological methodology. And then we were actually talking about the biological model of behavior and its components. And at that structure, I was especially talking about gene-environment interactions. And for the gene-environment interactions, I introduced that in the context of two examples. Do you remember what are those examples? Yes, the first example was monoamine oxidase A gene variation and its modulatory effect on the impact of environmental adversity and antisocial behavior, right? That was the first topic. And the second topic that we discussed in the previous class as an example of the gene-environment interaction is the effect of environment on the intelligence of maize-bright and maize-dull rats. As you remember, when actually we have the maize-dull rats and when we allow them to be grown in an enriched environment, it was found out that those maize-dull rats perform almost as good as maize-bright rats. So these two examples show us how genetic and environmental factors interact on the selected traits in behavior or cognitive capacities. So now, is there any other way to enhance the cognitive capacity of mice? What do you think? Do you think is there any other way to increase the capacity of the cognitive capacity of mice other than these kind of studies? Like what was the first study? In the first study, there was, gene environment, in the second study of maize-bright and maize-dull rats, there was a selective breeding, right? So the mice were selectively bred according to their cognitive capacity. So to achieve the most successful breeding or generation of the cognitively talented mice. And then different breed of mice were actually grown in different environmental conditions to assess and to modulate the effect of the environment on the capacity of the mice. So do you think there can be any other way to change the cognitive capacity of mice? What do you think? So let me give you a hint. There are two factors, genetic factors, environmental factors. Yes?
Maybe we can genetically modify that.
Exactly. Yes, maybe you can say it again so that everyone can hear you. Can you just...
Maybe we can genetically modify rats.
Yes, so by genetic engineering, right? For example, you can genetically modify, as we already studied in the case of selective breeding. Selective breeding is a very old style methodology. But with the genetic engineering, you can do a very direct manipulation of a target gene. And do you think one can produce genetically engineered mice that could be smart in these kind of maze tasks? Yeah, that was done already. And that mice is called the smart mice, which was published in 1999 already, so long ago, quite a long ago from this time. So what they did in order to, so that is very technical, and of course this is of course the details of this slide requires a background in molecular biology and genetic engineering, so I will not go to there, but I just want to explain you very briefly. What they did is, there is, we are already talking about genes and we know what are the genes. So, with the previous findings, they were thinking that maybe there is one gene called NR2B. NR2B is a subunit of NMDA receptors, which are actually brain receptors that are sensitive to glutamate. So these receptors are thought to be involved in learning and memory. Because of that, and that result was obtained because of previous studies, because of that they thought, what if we just increase the number of NR2B receptors in the brain, in the specific regions of the brain of mice? in the live organism. What will happen? What will be the consequence of that? So that was a study. They have chosen NR2B gene because it is very interesting. These NMDA receptors have two subunits. One is the NR2A and the other is the NR2B. And NR2A and NR2B and their production or expression in biological terms in the brain cells are developmentally regulated. What does it mean? That as you grow up, the NR2 actually A increases and NR2B decreases. So people were suspicious that as the NR2B decreases, our ability to learn may decrease because the level of brain plasticity, the adaptability of the nervous system to the environmental stimulation decreases. Okay, and maybe there was such a correlation with the, increased learning capacity with the increased amount of NR2B subunit, and that was actually in the adulthood that is decreased. In the fetal, in the earlier developmental stages, it is more. Because of that, they have generated a mice. This is how they generated, and this is they show the results. I will not go too much detail, but this is here they explain what they did. So they used a specific genetic construct which allows the expression of NR2B gene in the brain. And then here they show how NR2B gene expressed in the different brain regions, more than the normal animal, normal organism. And what they have found, as they already proposed, that they have found that there is an increased cognitive capacity assessed by the tests such as enhanced novel object recognition and hidden platform motor maze task. So these experiments already show that you can also increase the cognitive capacity at least for this case, for this organism, by selectively targeting specific genes. Okay? And of course, when this result was published, this was immediately taken very seriously because that brings the idea of gene therapy in humans, and also it makes a new dimension in the discussion of the heritability of IQ in humans. What would be the case in the humans? What would be, if we have people with cognitive disabilities, maybe we can actually solve these problems by gene therapy. What is gene therapy? This is a very little short explanation of a gene therapy, but gene therapy basically is a methodology by which you use viruses and you insert specific DNA sequences that you want to introduce into the organism. For example, NR2B encoding gene. And you put it into a viral construct which actually made not toxic or not dangerous for the organism, okay? And then you introduce to the specific tissue and allow the virus, while maintaining its own mechanisms, you allow the virus to introduce that gene of you into the target cell of the target organism. So you can basically, the idea of gene therapy is that you can introduce any gene in a specific tissue in the human body. It could be brain tissue, it could be any kind of tissue in the whole body. And that would allow you tissue-specific gene therapy and gene expression and control of the tissues and their physiology. Okay? So that idea of the NR2B overexpressing smart mice immediately bring the idea of generating maybe, virus gene therapy strategies for the treatment of associated cognitive disorders. And the second idea, of course, the issue of the heritability of the human intelligence, because I already described you the cases of the human intelligence in animal intelligence, but what is the case for the human intelligence? It was very debated and still under a debate, but we already know that there is a strong genetic component according to the twin studies. So because of that, so when this paper was published, immediately it became headlines in the news at that time. And one is actually, time cover. in the Time magazine, it was shown as the IQ gene. Because when you overexpress the NR2B gene, then you achieve animals that are smarter, smart mice. So this NR2B gene actually can be used in the gene therapy to make people very smart, very good in certain skills. I would say that this is a good example of exaggeration of scientific findings. Of course, the way how science works and how media works is very different. And maybe sometimes science needs such a makeup to take attention. Maybe it was even done by purpose by the authors of the paper. That is the IQ gene. We were able to generate, by playing the genes of the mice, we generated a transgenic mice, which is actually, smarter. So that could be possible for humans, designer babies, to generate smarter organisms. So, but in reality, as I said, that was just an experiment that gives a lot of insights for us to understand how genes, actually, not in this case, but in general, with the interaction with the environment, are involved in behavioral traits, including IQ. Okay, so, but not something to take it directly as an example that NR2B is an IQ gene. Okay, why? Why I want to mention that? Because I actually want to mention that in a very specific way. We don't actually think of behavioral phenomenon in terms of one gene. Okay, there is one gene and its variation, and if you, according to the variation you may have, and according to the environment and the interactions, you may have a certain behavioral manifestation. No, we don't think like that. On the contrary, we think that there are a collection of genes, and every gene has its variation, so each individual have a collection of genetic variation. And that means that this is polygenic, so not one gene. Okay, polygenic. But of course, in the former times, it was not easy to, there was no enough methodologies that was helping us studying all different genetic variations at once. So people were just first, focusing on small things that have a very strong hypothesis behind of that, according to the previous studies. That's why they focused on the monoamine oxidase A gene. They focused on the NR2B gene. But we know that actually it is not one gene, one trait, attribute when you think of behavior. It is multiple genes, sometimes 1,000 genes, and their genetic variations. And think about the complexity of the environment as well, and all together come together. So coming back to our discussion for the monoamine oxidase gene variation, I think you understand already. So if somebody, let's say I genotyped myself and I found out that I have the risky version of monoamine oxidase A gene, and I have also childhood abuse, my God, no, but, so do you think I should be suspicious that I could turn into a psychopath? Let's say I'm a male also, because it is more probable in males. No. Because probably you need also other genetic variations at once. That's why they work with so many big sample size, like 1,000 individuals, and they try to catch a relationship. Okay, so you always have to think behavior in terms of genetic variation of maybe thousands of genes and environmental interactions. And that is one of the reasons why it is so complex and why it is so hard to understand. Here, this smart mice was generated by a methodology called transgenic animal, transgenic organism. Transgenic organism is a genetically modified organism by the techniques of genetic engineering, by which a gene of interest is inserted into the genomic or genetic material of the organism. But that is not the only way to generate genetically modified organisms. So I don't want you to know all these details, but I just want you to be familiar with these terms. So far, I mentioned only transgenic mice. But the second example is actually here, is knockout mice. So when I talk about knockout mice, you should understand what I'm talking about, which is different than transgenic mice. In the knockout mice, which is a very actually typical strategy in biological methodology, we want to understand function of something and we just delete it and we look at the outcome, what happens if it doesn't exist there. So same for the genes. You select the gene and you knock that gene out from the organism and you generate a new organism without that gene and you look at the behavior. And that behavior is also studied in different levels of complexity, such as in the cellular level. What happens in the cells which do not have those genes? And then in the tissue level and in the physiological level, you introduce some electrodes and you measure physiological state of the cells by microelectrodes to see what happens to those cells when you don't have that gene. And then you also look at the live animal and you put them into some specific maze paradigms to test their cognitive skills or whatever the skills that you are studying according to your hypothesis. And based on that, you look at the result. So these kind of studies are typically called loss of function studies. So you want to understand the function of something, so you want to delete it. So that's why knockout mice technology in neuroscience and in also other fields. First time it was done in immunology, as far as I remember, enabled a lot of progress to understand all these questions. That's why I wanted to mention you this, but I will not go in detail. Today we have genome editing technologies. Before, for example, when I was a student, in our times, it was done with a different methodology which is called homologous recombination in the embryonic stem cells. But nowadays there are some other methodologies that emerged as a result of the technologies of genome editing. So that's it, and this is the detail how do they generate a knockout mice, but I will not go there. But I want to tell you two important things. So far, the only two genes I discuss, right? The first gene is the gene encoding for the monoamine oxidase A enzyme, right? And the second one is the gene which is encoding for the NR2B subunit of NMDA type glutamate receptors. So what do you understand when I say like that? So that means there is a receptor, okay, that is glutamate receptor, its name, all right? And it has some subunits, and one of the subunits is an NR2B, and I explained the significance of the NR2B. The expression of NR2B decreases as we approach to adulthood. So decreasing the ability to learn, probably. That is the proposition. So what if we recover that? What if we introduce more NR2B? And you saw that they were able to generate smarter animals. But, as I already said, in reality, it's not just one gene, okay? These are very simple and rather straightforward. That's why, and well-known, very well-known studies, like textbook examples. That's why I focused on these to explain you. But actually, in reality, you have to deal with thousands or millions of genetic variations to study, for example, psychopathologies and environmental interactions. Okay, so that was just the continuation of our previous introductory class. Now, in the second part of our class, today we are going to have some more technical, because if you don't know these technical parts, it's hard to understand what's going on. Okay, so that's why we will start with a neuron, its architecture or morphology. I like the term architecture, but morphology may be even more sophisticated than architecture, and its properties, features, functions, types. Here you see actually neurons that are stained with a technique called Nissil staining. Okay, and it's very, I think probably you have already seen how the neurons are seen, right? Some of you already know, with a very typical shape, different than any other cell. Okay, but when you actually do this Nissil staining, it doesn't look like that. So in the former times, more than 100 years ago, when people used Nissil staining technique, they were observing the neurons under the microscope with this shape. Later, there was this opportunity of the Golgi staining, which I was explaining already with the drawings of Ramon Cajal and how he was observing the neurons under the microscope. So different methodologies allow you to visualize the same thing in a different way. So here, when I was the other day reading, I was so impressed and I wanted to share it with you. So I want to read what Ramon Cajal, when he was actually studying the neurons with the Golgi staining under the microscope and drawing, which led him to get Nobel Prize, writing about his impressions. Let me read it loudly. Against a clear background stood some black threadlets, some slender and some smooth, some thick and thorny in a pattern punctuated by small dense spots. All was as sharp as a sketch with Chinese ink on a transparent Japanese paper. And to think that this was the same tissue which, when stained with carmine or logwood, left the eye in a tangled thicket where sight may stare and grope ever fruitlessly, baffled in its efforts to unravel confusion and lost forever in twilight doubt. Here, on the contrary, all was clear and plain as a diagram. A look was enough. Dumbfounded, I could not take my eyes from the microscope. So he was, right in one of his books, he was describing how actually with the Golgi staining technique, he was able to see this very clear, according to him, because that structure allows him to predict how these structures work, because structure and function have a close relationship and correlation in biological systems. No structure is just for nothing. There is a reason. So, and here now we have the prototypical neuron, where you have to know some very basic structures of the parts, structural parts, and one of them is the soma, which is the cell body of a neuron, okay? Soma is very important because there you have the nucleus in which you have the genetic material that are constructed as chromosomes. They are found in the form of chromosomes, okay? And then you see two different classes of processes extending from the soma. One part of these processes are called dendrites. Okay, dendrites. And the other is rather long, is axon. So we give different names to them because they have different functions. Dendrites receive signals from other neurons. An axon sends signal that is coming from dendrites, and then there are some actually mechanisms here, but after that, if it is okay, then that signal is going to be sent by axon to the other neurons by which this neuron is being connected. Okay? So what you have to understand that neurons have some basic structures, and one of them is the dendrites. By dendrites, the neurons receive signals from other neurons. And by its axon, it processes all the signals that is received by dendrites, and after the processing, which is a topic itself, so we are not going into that level in this introductory course, but then those signals are sent to the next neuron through its axon. So dendrites receive the electrochemical signaling from innervating neurons, and axon is sending that signal to the next neuron that it is connected to. Is that clear? So, but here I would like you to focus on one thing. If you look at this one neuron, and if you look at its dendrites, Do you think how many different neurons can be connected to this neuron with its dendrites? 10,000 maybe, depending on the neuronal type. Okay? So think about that neuron as a information processing unit that is somehow taking 10,000 different signals in a very high spatiotemporal scale. That means that is both according to the three-dimensional location and time that neuron is able to process all this information. Okay? And then the whole information is being actually finally processed at this specific location, which is called axon hillock. That is also another important thing maybe you would like to write on your notebooks, where all the signals that are received through the dendrites are finally processed in the position of axon hillock. Because then after that, all these information have to make a final meaning, have to come a final conclusion. Then axon can send it to the next neuron. Okay? Yes?
Could you repeat, please, how this area is called?
Axon hillock. Yes, we will have more chance to repeat them during the seminars also. Hillock. H-I-L-L-O-C-K. So, but you know, actually it was already very hard for scientists to see that neurons with this very complicated structure were able to actually operate like other cells. And that was actually the study of Cajal and other scholars led to the idea that actually neurons stick to cell theory. And that was eventually called as Neuron Doctrine. But before that, it was not clear whether the neurons are sticking to cell theory or maybe neurons are continuous, like a reticular, like, you know, veins surrounding the whole body. They were thinking that in the brain, the nerves also like that, like a web. Okay? And there was actually, this is one of the debates in the field of neuroscience that was like debated by two scientists. So Golgi, who developed the technique of Golgi staining, actually proposing very strongly the idea that neurons are continuous like a reticulum or a network similar to arteries or veins of the circulatory system. On the other hand, Ramon Cajal, using the same technique that is generated by Golgi, so they use the same technique, but two scientists working on the same substrate, but they have opposite conclusions. So you see, this is a very good example, actually, how doing the same things you may end up with the different conclusions. But today now I mean, nervous system is so complex. If I would like to speak in the maybe very beginner level, I would say that we prove that a lot of studies have found that actually neurons are discontinuous because they are not, they are actually disconnected and interact with each other by specific subcellular structures which are called synapses. But on the other hand, later on, it was discovered that there are some specific structures which are called gap junctions. And they are actually junctions that allow direct flow of the ions between the two neurons. And that also make you to idea that actually then that is another attribute of the neurons, that neurons may be continuous if you think of in terms of the gap junctions. So maybe they are both discontinuous and continuous. So, but let's don't go there, and let's assume that, especially because at that time they were not sure if the synapses were really able to make the neurons structurally discontinuous. But today, with the advancements in electron microscopy, we know that neurons are discontinuous in that aspect. So if somebody is asking you, are the neurons continuous or discontinuous, maybe you should respond them after taking this class. What is your... It depends from which point of view you are asking. Are you asking in terms of synapses? Yes, they are discontinuous. Are you asking in terms of other structures that make direct contact? No contact, just, you know, open junctions between the neurons, then I would say that they are continuous. So it depends on your criterion. But now we are in a completely different landscape. This is a confocal microscope, okay? And confocal microscope allows you to work with the neurons in the live cell conditions. Or for example, you isolate the brain of an organism and you make very thin slices, and that slices under specific physiological conditions, they can stay alive for 7 hours or so, okay? And then you can actually work on their structure, but not only structure, but also physiological state under the live cell conditions, the interaction of different molecules that are found in these cells. You can work on that topics, on these kind of topics, with the use of these kind of specific microscopes called confocal microscopes. These microscopes are so advanced that, for example, you use genetically labeled systems. So because it is very hard to study the nerves in so detail, or any kind of cell, it's so complex. You have physical barriers, you have chemical, biological complexity. You want to just focus on one kind of molecule and its specific interaction with another kind of molecule. And how are you going to differentiate those from the rest? Because there are thousands of different kind of molecules operating at the same time. So we use genetic engineering to actually genetically label with fluorescent colors our target molecules, and in the live tissue we can study their functions. And in that aspect, chemical and physical sciences are being integrated. So advancements in the optics, microscopy, or chemical methodology such as, fluorescence. And it was already 1990s, for example, two-photon microscopy was developed. And then that was another progress. Why? Because that allows you to study live organism. Because, you want to, how are you going to study the cells of a live organism? In the beginning, you isolate those cells, and then you put those cells into a slide, and you observe. And you don't want cells to be, degraded. That's why you fix the cells. And these are actually dead, frozen cells. Okay, that was done. But then the next stage, okay, I want to study live cells. I want to see when the cells are living. I want to observe. Okay, that was done, as I explained. But now I want to study the cells of a living organism in the real time. So, for example, but how can you do that? I don't want to, I open the skull, I remove the membranes of the brain, and I keep the intact organism under a microscope, and I want to have an optical system that goes to the layers of the brain without actually really in reality destroying that. So you want to go maybe 10 micrometer deeper into the brain tissue on the upper part, but without destroying the tissue. So that kind of studies or developments were allowing us study that phenomenon in such an excellent state. So now you can imagine what kind of opportunities are available today. Here is a very textbook example of the different levels of magnification. If you can see here, for example, we are in this scale. So less than 100 micrometer. So the microcosmos. So many different, the scale of the light microscopy is starting from the 0.5 micrometer, okay? So you can go down to the 0.5 micrometer as a resolution. But I have a now different view for that. And it is because of that. So the Nobel Prize in 2000, I think, when is the Nobel Prize is not written the date, but 2014, I guess, or 17. But yes, 2014, yes, I didn't see. So the Nobel Prize for 2014 was given to these guys because they were developing optical microscopes that are actually having much increased level of resolution. So this example now have to be, this is a textbook example, but it has to be updated. Why? Because now even the microscope is now in the range of nanoscope, because it is more than, it is almost, it is now working in the resolution level of electron microscope with the current advancement in optics. Okay? So I think it is important that now what kind of advancements are existing and how they can be studied in order to understand neurons, their molecules, their interactions, and the physiology, all actually at the same time. Because that is not something you can differentiate, you can separate and study. That was a typical actually methodology in the former centuries, but today actually you have to, it is our human brain to separate the things because it is less complex that way and we focus on one thing. That is our tendency, but things are not, behavior is not just behavior you observe. There is a physical substrate behind of that, and it is made up of by the activity of these neurons in very different levels of resolution and complexity, and that starts from atomic level to systemic level. So somehow we are looking for an opportunity to work all these different levels of complexity at the same time and to find a useful relationship between them. And there were some research methodologies doing that. But that is not our subject today, but all I want you to know, what is the required landscape in order to understand behavior. Now, this is an image of confocal microscopy, and now I would like to talk about some basic principles of neurons and how they operate. So, brain information processing is mediated by excitatory and inhibitory signals between neurons. Those excitatory and inhibitory signals are generated by specific molecules, signaling molecules. We call them as neurotransmitters, such as glutamate, GABA, and glycine. And those signaling molecules act or work in the specific subcellular structures, which I already mentioned in the previous class, called synapses. Of course, a synapse cannot be just say, okay, there are two extensions here are overlapping, so they are synapse. No, we have to verify it by molecular markers if it is a real synapse or not. But just as a symbol, I am showing you. So it's a subcellular structure, okay, between two neurons. It is a subcellular structure at which the signal transmission between two neurons take place. And it's a subcellular structures by which it was proposed that neurons are discontinuous, as we already discussed. So that's why it is important for you to know the function of the synapses. But before that, I want to show you a very basic feature of a neuron. So think about a neuron. Think about that you have them, in a live slice preparation with this advanced microscopy techniques, and think that you have microelectrodes and system that allows you to measure the electrical state of the neuron. So they did that. They are doing that with some methodologies called electrophysiology, and they have found that neuron at a certain state, which is called resting state, which I will explain you later, has a potential difference which is minus 65 millivolt. So they inserted an electrode, okay, And then they measure that there is a minus 65 millivolt potential difference between the inner and outer part of a neuron. Okay, because of that, we call the neurons as polarized cells. There is a potential difference. Okay, and if you find a neuron and that is called resting state. What does it mean, resting state? I tell you now. That means neurons is at rest, sleeping, very tired. Okay? So at rest, neurons have a physiological state, electrophysiological state, which is shown as, measured as minus 65 millivolt. And any kind of, so take this minus 65 millivolt as a reference point always for the rest of the class, because I will use certain terms related to that. If I say a neuron is excited, that means that this minus 50 millivolt is going and becoming minus 30 plus 10 millivolts, going through the positive part, positive scale. Okay? If I say neuron is inhibited, that means that this minus 65 millivolt is becoming even more negative, minus 80 millivolts, for example. Okay? So minus 65 millivolt is our reference point, and it's the reference resting state of a neuron. Now I would like to tell you another important term, excitatory postsynaptic potential. Okay? EPSP. And the other term is inhibitory postsynaptic potential. IPSP. All right? So if this minus 65 millivolt, if it becomes minus 50, becoming less negative, it is caused by excitatory postsynaptic potential. So first this, then I will show it in the figure. Just keep it on mind. If this minus 65 millivolt is becoming minus 75, that means there is an addition of inhibitory postsynaptic potential. That's why we measure it more negative. Now think of a neuron that is having both excitatory postsynaptic potential, shown with red colors here, okay, and inhibitory postsynaptic potential, shown as blue color. These are representing other neurons. Okay, other neurons, I was lazy to draw by myself. This is from textbook, but I modified it a little to explain. Okay, so that means there is one neuron, and that neuron, if it interacts, is supposed to give excitatory postsynaptic currents, which will be observed as excitatory postsynaptic potential in the receiving neuron. And if you look at this one, this is just random, of course. In the real systems, it is tightly regulated. Which neuron is connected with which neuron in which way. It is under genetic control and modified with environment, by the way. They show it in the experiments. But here it is random, I'm showing you. So if you look at, for example, this one, if it is, as blue shows, inhibitory postsynaptic current, is actually innervating this neuron, and inhibitory postsynaptic current is introduced to that target neuron. This is our target neuron. Okay, and that target neuron is going to have an inhibitory postsynaptic potential. And and this is not constant. The way how many innervations are being made into a neuron and which of them are sending these signals at which time is very variable and complex, and it has a very short time scale. So anytime, look at how you now think, perceive. process all the information coming from your environment now. This is all now working under this kind of principle with billions of cells at the same time. And I'm just explaining just one cell. And that cell is actually by itself is a processor, because it has to, every time, it has to cumulatively collect all the total iPSPs, total EPSPs, okay? They are all sensed by specific receptors that are found in the cell. It's a physiochemical process. And then that is the way they all together come and meet at the axon hillock. An axon hillock has sensors. Think of a thermostat, maybe. So it has its own very sensitive sensors, and it just decides, oh, is this cumulative, the total iPSPs, total EPSPs come together, and it should just pass over a threshold. If it passes that threshold, then this neuron is going to send a signal to the next neuron via its axon. Okay? So this is how a typical neuron actually decides whether it is going to send a signal or not to the next neuron. And all the principles that I applied here, explained here, is also applicable for this arrow, which is actually representing another neuron. If this neuron is sending the signal to this neuron, that means that, what does it mean? That means that it already actually evaluated and processed all kind of innervated signals coming to that, and processing that information and sending or not sending information. If it's sending, that sends either could be an iPSP or EPSP, depending on the identity of the neuron. So for that now you will learn that there are different kinds of neurons, and among them you can maybe say that there are excitatory neurons and there are inhibitory neurons. Inhibitory neurons are those, if they are actually stimulated enough, they will send inhibitory signals to the neurons to which they are targeted to. And that inhibitory signal is this iPSP. Anything that will make this more polarized, more negative. An excitatory neuron is the other way. An excitatory neuron is a neuron which is going to stimulate the target neuron, that it is going to have an excitatory postsynaptic potential, meaning that this will become more positive. And what do I mean by more positive and more negative? What do you understand when I say more positive and more negative? Okay. With the system, you measure it, but what does it do? How is it established such a way? It's just ions. What are the ions? These are molecules. These are some atoms that either gained or lost one of its electrons, then they are charged. These are ions. So there are positively charged ions, there are negatively charged ions. In the environment in which the neurons are found, it's like a soup of ions. Okay, we are all our tissue is embedded in water, but that's not pure water, right? There are so many different molecules and ions that is found. And for our context, we have to focus on 4 ions. These are, actually, I will show it here. This is the cross-section of the membrane of a neuron. And here is the zoomed version of that. This is the cell membrane. And inside of the neuron, there are more negatively charged ions. And that makes a kind of negative charge accumulation in the inner surface of the cell membrane. And in the outer part, there are more positively charged ions. And it is shown like that. Because of that, inside of the neuron is more negative at rest. And if it is even more negative than the rest, it is likely to be inhibited. If it is more positive than the resting state, it is probably, because these are more complex than just I explained, that's why I use more, it's probably excited. Okay, so here it is just shown as actually charges, negative charge and positive charge. But what are those charges? These particles that are actually involved in the maintenance of this potential difference is mediated by the electrochemical balance of major ions. These ions are sodium ions, which are positively charged, potassium ions, chloride, and calcium. Okay? So all these ions, and depending on the permeability of the cell membrane of the neurons to those ions, there will be an electrochemical balance established each time. And what determines the opening or permeability of the ions to the cell membrane? So cell membrane is a barrier. It's a barrier, and it has some specific channels inside, and those channels are sensitive to selective ions. So there are channels for calcium, there are channels for potassium, there are channels for sodium. But those channels are not always open, they are not always closed. They are actually controlled by these systems when they are open and when they will be closed. But I want to now show you especially the structure of the cell membrane. So this is the soma here, the soma. And if you take a section from the soma, this is, you see here, the structure of the cell membrane. And the cell membrane is made up of a structure called lipid bilayer. So this is a structure that are, it has such a chemical structure. I will not go too much into detail of chemistry, but what I would like to say that it has such a structure, chemical structure, that, for example, these parts are not in tendency to, they do not tend to interact with water because of their chemical structure. It's not favorable according to chemical principles. That is an observation. It's a chemical observation. On the other hand, these parts, these heads, okay, they tend to interact with water. So if you take these molecules and put it inside the water, they immediately make such an orientation that their parts, which are not to interact with water, they somehow escape from water, and they make their inner part to be, so these, parts that are not in tendency to interact with water, they will escape from water, and they somehow find a way which best orientation allows them not to interact with water. They will get oriented that way. And that is the way actually how cell membranes are made up of phospholipid bilayers. So, and that is the even bigger version. So those parts here which tend to interact with water, they are called polar. Polar is very basic term, but if you even want to study simple psychopharmacology, maybe you need to know what polar is. Polar is a molecule that has its charges unevenly distributed. It could be a neutral molecule, but the charges are not distributed evenly. Okay? And that polarity allows it in a way, or makes it in a state that it can interact favorably with water. And these kind of molecules are called hydrophilic. I think you should know it from high school. Do you? Some. I don't know how in Russia, maybe you have different sections, but science student, I don't know if you have science section in high school and social science section in high school, or it is unified. If it is unified, you should know if you take some chemistry or biochemistry, but I will learn Russian system soon. But to my knowledge, this is high school background. So, okay, so I would like you to know one important feature of the cell membrane of a neuron, which is typical cell membrane of any cell, okay? And that is, it is made up of phospholipid bilayer. It has polar and non-polar sides. The polar sides are actually extended toward the surface from the intracellular and outer part of the cell. And non-polar sides are just those that are found in the inner region of the bilayer. And that is determined because of their preference or chemical preference or stability according to interaction with water. Okay? That is something very simple and important fundamental characteristic of the cell membrane of a neuron. Another important thing that you need to know, especially if you want to understand where do all these EPSP and IPSPs are coming or do come. And that is these cell membranes have receptors embedded to them. Okay, this is one receptor, but actually you have to think a cell membrane a mosaic of different receptors. And where do those receptors come from? Genes. Genes produce them. Okay, genes produce different repertoire of proteins. So the protein repertoire that's found in a cell is dependent on the activity level of the genes that is found in that cell, and that is not the same in any cell. Every cell has its own protein repertoire determined by the activity of the genes. So this is now a good example when I talk about NR2B subunit of the smart mice. This is exactly what I want to say when I talk about a subunit. Because proteins, in this example, it's the channel protein or receptor found or embedded in the cell membrane, typically are made up of by subunits. Look, 1, 2, 3, 4, 5. Each of them are different colors. That means, and they are 5. That means this is a heteropentamer. It has 5 different subunits. So a protein is actually assembled from a group of subunits as a peptide, and then it folds and gets its final 3D structure. Only after that it does its job. And normally each of these subunits are produced by separate genes. Okay? So when I talk about a subunit, you have to understand that there must be a gene in the cell producing that subunit. And that subunit is a part of a bigger molecule. Why did I show that? This is DNA, messenger RNA, and protein. So this protein here, in this example, you have to take one subunit here in this example, and you have to think that that subunit is actually has its own gene transcribed into messenger RNA. It takes place in the nucleus. Nucleus is the part of the cell in which the chromosomes are found. And the chromosomes are transcribed into messenger RNA. Which chromosome part is going to be transcribed into which messenger RNA is a very complex process, and it's not the same in every cell. Every cell we have the same set of genes in the brain. But not all genes are actually so the genes are turned on when you have this flow. The genes are turned off when this does not happen. This is called differential gene expression. Okay, so every cell, we were talking about billions of cells in the brain. They have the same genes, but those genes are not active at the same way. It's differential. So think about 20,000 genes on each neurons. But here I wrote as protein, but actually if that protein is made up of some subunits, then you have to think of actually just one subunit is made by one gene expression and another subunit, and they assemble after that. Okay? This is very simple, just to see the relation between gene and protein. So when I talk about protein, that means we are talking about their genes as well. Okay, this is very important now because you know we were discussing the neurons in terms of their, in terms of what? In terms of their structure, in terms of how they were interacting with each other. And they can be classified according to their, number of dendrites, specific anatomical features. But another dimension of their classification is actually shown in this slide. And that is when you have, for example, this is the brain of the first person, and this is the brain of the second person, okay? And so this is, of course, they cannot just get your brain and extract your tissue, right? But these are postmortem brains. And then this, do you remember this DNA messenger RNA protein gene expression? So for us, messenger RNA isolation is important, is an important signature to see which kind of genes are expressed, which genes are not expressed in a given tissue. For that reason, they compare, for example, the messenger RNA from one person extracted from one brain, and the messenger RNA of another person extracted from another brain. And they compare which genes are differentially expressed differentially in the brains of these people. Okay? And that is done by a technology called gene chips. And you can see how all these so many different things are differentially expressed and found. And you can characterize the brains of the people according to molecular signatures. Here, as you see, gene with reduced expression in brain 2, gene with equivalent expression in both brains, gene with reduced expression in brain 1. And this is you study in this with a slide, and this is a specific, I don't want to go in there, but this is something like a sequence. You do a method called hybridization, and that allows you to see all these differential factors between the molecular profile of these two different brains belonging to different people. Okay? So you don't need to go to this technical background, but all you have to understand that you can just compare messenger RNA level of the brains of different people or any tissue. They also diagnose cancer based on that, for example. They do that in any tissue and as well to the brain tissue. And you can classify, compare, contrast which genes at which level are being expressed in different people. And now there will be some emphasis on understanding of the basic principles driving the formation of the balance in the cell at rest, neuron at rest. So what are the factors? I already mentioned that there are ions. I already mentioned that those ions are not equally distributed across the cell membrane, that there is more negatively charged ions compared to the outer part of the neuron, and that actually leads to the minus 60-fold millivolt potential difference, right? That is clear. But what are the factors driving that minus 65 millivolt? How it becomes like that? So there are two important features that is making a neuron having such an electrophysiological state, and that is determined by two factors: the electrical gradient and the chemical gradient. Okay? What is chemical gradient? Now, as it is shown in this example, think of a cell, and this is the cell membrane, okay? And assume that the cell membrane is not permeable. It's not permeable. The sodium ions are more concentrated at this part, and the sodium is going to diffuse to the second part. Fluor ions, or negatively charged ions, are more concentrated at this part, and they will diffuse into this part. Until when? Until their concentration, not amount, that the students sometimes are missing. It is all about disorderness in a given volume. Okay? Amount doesn't mean anything. What is the volume? And then you have to decide on concentration. So the concentration of each of the ion is becoming same at two parts of the membrane. Then there will be still, do you think the passage of ions are going to stop? No, they will not stop. These systems, they never stop. They are always in activity, but the net movement of the ions across these channels is going to be zero. And that is when there is an equilibrium state is established. But in this example, there are two kinds of ions and there are two kinds of channels that are selective to each of these. But in the neuron, at rest, it is not like that. There is an already unequal distribution of ions across the cell, and their permeability is not equal. At rest, mostly, for example, potassium channels are open. And the potassium is going to go to the cell or lead to the cell according to its concentration gradient. But there is another factor, the electrical or electrostatic interactions. These are charged molecules and ions, sorry, they are not molecules, they are ions. These ions have also charges, and because of these charges, they will have an electrical interaction between them. And that will be also another factor that will determine the movement of these ions. For example, think of this case. So sodium and chlorine are going to attract each other to some extent, but they are dissolved in the water. Whose dissolved state in the water is thermodynamically more favorable. That's why they are dissolved. We can discuss for an hour about that. But that is all explained under a logical, chemical, or thermodynamic principle. But that is, they are dissolved, but there is some electrostatic interaction between them. But there are many, these red dots, each of them represent sodium ions. And there will be also a repulsion between sodium ions as well, right? So the sodium will go here, and these sodiums are going to, do not attract each other. There will be attraction between also if chlor and sodium here a little. So this is just an example to see how many different diffusional and electrical interactions can take place at one time. And then eventually there will be a balance that is established according to these factors. And that is minus 65 millivolt according to the concentration and electrical gradient of the potassium ion, to which the cell membrane permeable makes the cell to have resting state as we define physiologically. Okay? So here, this is the neuron, and this is the inner part of the neuron. It looks like a pipe here, but actually it's not a pipe. The inner part. So what is the concentration of each of these ions at the inner part and outer part of the neuron? Can you read? So what is it in the inner part? What is it in the outer part? And let's do not focus on the actual absolute numbers, but let's concentrate on relative numbers. Which one is higher? So inside of the neuron, concentration of potassium is higher compared to the outside. Inside of the neuron, concentration of sodium ion is less compared to the outside. Inside of the neuron, concentration of calcium is less compared to the outside. Inside of the neuron, the concentration of chloride is less compared to the outside. So there is only one ion that is more concentrated according to the outer part of the neuron, and that is potassium. Okay? So assume that the cell membrane at normal resting state is only permeable or mostly permeable to the potassium ions. So only the factors driving the potassium equilibrium is going to determine the membrane potential of that neuron. And that minus 65 millivolt is determined according to that. You can calculate it by a special equation called Nernst equation, but I will not go to that deep today. If you want, I can another time. Okay, so I think today we learned some basics. I want to make a summary, and I hope it will be more clear for you. Neurons are specific polarized cells, and that's polarized cell meaning that they have a potential difference, and that is measured as minus 65 millivolt at rest. That means that that neuron is not sending any signal. That is the first thing you understand. That resting membrane potential is mostly established by the equilibrium or electrochemical balance of the potassium ions because of the reasons I discussed. Okay? And that balance is interrupted when? Do you remember? For example, let's take this neuron, and this neuron is actually innervated by other neurons, and it receives excitatory postsynaptic potentials, or it actually accumulates excitatory postsynaptic potentials and inhibitory postsynaptic potentials from other neurons. And those excitatory and inhibitory postsynaptic potentials have the ability to manipulate the membrane potential above the minus 65, below the minus 65. And that will make a neuron to give, that will contribute a neuron the probability to send a signal to the next neuron or not, which will be processed in the brain, in the neural region called axon hillock. Okay? And any excitatory postsynaptic potential, here, why did I actually mention these? If you read some popular psychology article, normally you read articles about dopamine, serotonin, right? Everybody knows serotonin, right? What do you know about serotonin? Sorry?
We're happy when we have serotonin.
And dopamine?
If I'm not mistaken, it's
Dopamine is for motivation.
and achievements.
Motivation, who said motivation? Yes, motivation and
Achievements.
achievements.
And yeah, as a result of
Okay, what else? Dopamine?
Anticipation.
Addictions?
Anticipation of pleasure.
Pleasure? Yes, addictions. Because that dopaminergic system in specific brain regions, when they are actually overstimulated by some drugs, then we have addictions. Okay, but I talk about these. Why? They are the actually main, so they are like molecules like dopamine, serotonin, okay, nothing else. But their role is more fundamental, like they are the main columns of a building. Think of it, look at these columns, how many columns we have. So the building has to have its, a very well calculated static balance with its columns and everything. If you have some engineering background, you would know. And that is here, the electrochemical process in the brain in that level is actually mediated by especially these two guys, glutamate and GABA. I already discussed with you what is excitation, what is excitatory postsynaptic potential, and glutamate is the one that contributes to that. So if a glutamate is released from one neuron, the other neuron is going to have excitatory postsynaptic potential. And the counterpart of it that has the ability to decrease the membrane potential down to minus, even more negative than the minus 65 millivolt. That is meaning that the cell is going to have an inhibitory postsynaptic potential, and that is mediated by GABA. Okay, so now everybody understands when I say glutamate is an excitatory neurotransmitter. Now you know the whole story. And the GABA is an inhibitory neurotransmitter. Okay, glycine is another inhibitory neurotransmitter. That's why I mentioned that. So the majority of the neurons in the brain are excitatory and glutamatergic. And you know, think of that excitation. If you cannot control excitation, you have a seizure in epilepsy. And that excitation has to be balanced with GABAergic inhibition. And that excitation inhibition balance is extremely important for our brain function during health and disease. Okay? Any questions? Yes?
It's about the potential
Microphone.
in neurons.
Yes.
Yeah, it's about potentials in neurons. Does the precise number of these potential matters, or is just like artificial neurons where 0 or 1, excitatory or inhibitory?
So excitatory potentials are not precisely like 0 or 1. Excitatory potentials are coming from all different neurons in different values with the values I already explained you. But the neuron itself receiving all these EPSPs and IPSPs, and that is the point at the axon hillock is going, whether the neuron is going to send an action potential or not, or signal. I didn't talk about action potential yet, but just to answer the question of your friend, that 0 or 1 state of a neuron is determined after all these different signals for a given time are being processed. If it is above a certain threshold, it is on, 1. If it is not, it is 0. That means the neuron is going to stay silent.
But it sends certain signal, and it's not. It has also not a 0 or 1, and this fluctuation of potential doesn't matter. Doesn't also send any message.
I didn't understand your question, but as far as I understand, maybe you want to ask that so this, for example, if a neuron receives a signal from another neuron, that will cause its postsynaptic potential become either excitatory postsynaptic potential or inhibitory postsynaptic potential. It's a value, and it will be distributed through the surface of the neuron, and it will come and migrate to the axon hillock. At the another part of a neuron, another events like that is going to take place, and it will undergo the same process. Another. They all together come, and there are specific sensors that are found in the axon hillock. Those sensors, depending on their biochemical structure, can be turned on or off depending on the total value that is accumulated as a result of the accumulation of all EPSPs and IPSPs that are coming at one time, at a given time. And if it is above a certain value, it will cause that neuron to send a signal through its axon. That's why the value is important. And that value is determined by the channels that are found in the axon hillock. And these are voltage-gated potassium channels, actually. But I didn't want to describe in detail. So the value is important at that point, if you were asking that. Or if not, I can explain you after the class. Any other questions?
Yeah, one.
Yes.
About gene therapy, is it already implemented?
Yes.
Yes. And it does not lead to cancer?
No, what do you mean by implemented? For example, when you say implemented, in medicine, you can make certain things as a proof of principle, but when it is really applied to the patients, require a lot of stages of regulatory stages, and that will take a time. Here, most of the time, I talk about the preclinical studies. Yes?
But how is it possible to target certain cells by viruses?
Yes, it is possible, my dear. Take a genetic engineering class from me, I will teach it. I wanted to open genetic engineering course in SAS, but I thought maybe this course is more interesting. That is another subject, but it is possible, Milana. And we have this technology, believe me.
Great.
Okay.
Thank you.
But if you really want to know, what I say that every gene has its promoter that allows it to be active in a given cell. If you know those promoters and which promoter make a gene actively expressed in which cell, and we know this, we just combine any gene with that promoter and we activate the gene. So it doesn't matter if you give it a virus or not. Virus just is a delivery methodology. Once the virus delivers the genetic construct into the cell, it is not belonging to virus anymore, and the rules of the molecular biology is going to take place, and that is just gene, messenger RNA, protein, and if it is in the right cell in the right moment, as we designed it in the beginning, according to these promoters and their interactions, as I explained, it is possible. So normally, virus, to take it to all cells, viruses cannot just, target only one cell. But let's say there are 100 cells that are delivered by the virus. There are 100 cells that were targeted by that virus, and virus introduced its genetic information together with our genes into that cells. But we designed our genes in such a way that those genes are going to become silent in the cells that we don't want to be active, because we designed them according to that idea. And that way we can control, even with the viruses, which genes are going to be active in which cells. Any more questions? Okay, if not, so today we learned the basics of neurons, and we have some more opportunity to study some detailed structures in the, to foster what we have learned in the seminar hours. So for those who do not have any background in these subjects, don't worry. In the seminar, we will fill the gaps. And but in the open courses, we just now, in the next course, we will go to another topic, mostly anatomical features of the brain. And that is not cells anymore, that is the structures. So which kind of structures we have in the brain, and which kind of functions are correlated with these structures. Okay, thank you.